targeted genome modification has been hampered by the lack of an efficient tool to create double-stranded break at designed location. The CRISPR-Cas system is an RNA-guided nuclease system for targeted introduction of double-stranded DNA cleavage. Originally discovered in bacteria as an acquired defense against foreign pathogen, CRISPR-Cas technology has been demonstrated to work successfully in all the common model organisms, making it one of the hottest technology breakthroughs in 2013. Targeted genomic cleavage requires two key elements: a homing device and an endonuclease. In the CRISPR-Cas platform, the homing device is guide RNA or gRNA, and Cas9 is the enzyme introducing the double-stranded cleavage. The gRNA contains a 20-nucleotide sequence linked to a tracer RNA scaffold. The targeting sequence is immediately upstream of a protospacer adjacent motif, or PAM. This is sufficient to direct the Cas9 nuclease to the complementary site in the genome and create double-stranded break. Compared to previous genome editing methods, such as zinc finger and TALEN, CRISPR-Cas is cheaper, quicker, and more accessible for researchers. Due to its amazing simplicity, CRISPR-based genome editing can be achieved with a simple transfection. Oragene offers an all-in-one vector P-Cas guide, engineered with all the essential elements for targeted cleavage. The codon-optimized Cas9 expression, a cloning site for 20-nucleotide target sequence, and a gRNA scaffold downstream of the cloning site to be transcribed into a complete gRNA by a U6 promoter. With a target sequence cloned into this vector, cells transfected with this constructs will express Cas9 nuclease and the guide RNA, which will then lead to sequence-specific double-stranded break in the genome. Other vector variations are also available to provide added convenience, such as GFP for transfection monitoring or lenti backbone for viral delivery. For researchers who perform mRNA microinjection or mRNA transfection, a T7-driven expression vector is available. With the CRISPR-Cas system, genome editing is now smooth sailing. Two types of editing are commonly used. Homologous recombination utilizes a repair template. The desired changes are flanked by left and right homologous arm sequences. Upon double crossover, the desired change is integrated into the genome. Applications include gene knockout, gene tagging, and site-specific mutagenesis. Non-homologous end joining, where the ends of the break are joined together, introduces random mutations, insertions, and deletions. This can also be used to create variant libraries. Applications for CRISPR-Cas9 are endless. You can generate an indel variant library at a targeted locus. You can knock out a target gene and simultaneously knock in a functional cassette, such as a mammalian selection marker or a fluorescent marker, and you can introduce pre-designed mutations. One common application is the promoter study. It requires the insertion of a reporter gene, such as luciferase and/or a GFP, at the first exon of a target gene. This results in knocking out the target gene and knocking in the reporter, therefore enabling the researcher to monitor the promoter activity. For this application, Oragene offers a complete solution. Simply search with a gene symbol and you can find a fully functional kit containing 2 gRNA vectors, 1 donor vector, and 1 scramble control vector. Another common application is the safe harbor insertion of transgenes. Compared to other transgene insertion methods, this method provides total control for insertion site, orientation, as well as for copy number. A ready-to-use kit for this application is coming soon from Oragene as well. Oragene brings you a complete CRISPR-Cas solution that is simple and easy to use. To learn more, please visit the Oragene website.